Humans are not the only species facing a potential threat from SARS-CoV-2, the novel coronavirus that causes COVID-19, according to a new study from the University of California, Davis. An international team of scientists used genomic analysis to compare the main cellular receptor for the virus in humans, angiotensin-converting enzyme 2, or ACE2, in 410 different species of vertebrates, including birds, fish, amphibians, reptiles and mammals. ACE2 is normally found on many different types of cells and tissues, including epithelial cells in the nose, mouth and lungs. In humans, 25 amino acids of the ACE2 protein are important for the virus to bind and gain entry into cells. The researchers used these 25 amino acid sequences of the ACE2 protein, and modeling of its predicted protein structure together with the SARS-CoV-2 spike protein, to evaluate how many of these amino acids are found in the ACE2 protein of the different species. Animals with all 25 amino acid residues matching the human protein are predicted to be at the highest risk for contracting SARS-CoV-2 via ACE2, said Joan Adamus, first author for the paper and a postdoctoral research associate at UC Davis. The risk is predicted to decrease the more the species' ACE2 binding residues differ from humans. About 40% of the species potentially susceptible to SARS-CoV-2 are classified as threatened by the International Union for Conservation of Nature and may be especially vulnerable to human 2. The study was published August 21 in the Proceedings of the National Academy of Sciences. The data provide an important starting point for identifying vulnerable and threatened animal populations at risk of SARS-CoV-2 infection, said Harris Lewin, lead author for the study and a distinguished professor of evolution and ecology at UC Davis. We hope it inspires practices that protect both animal and human health during the pandemic. Endangered species predicted to be at risk. Several critically endangered primate species, such as the western lowland gorilla, Sumatran orangutan and northern white-cheeked gibbon, are predicted to be at. Other animals flagged as high risk include marine mammals such as gray whales and bottlenose dolphins, as well as Chinese hamsters. Domestic animals such as cats, cattle and sheep were found to have a medium risk, and dogs, horses and pigs were found to have low risk for ACE2 binding. How this relates to infection and disease risk needs to be determined by future studies, but for those species that have known infectivity data, the correlation is high. In documented cases of SARS-CoV-2 infection in mink, cats, dogs, hamsters, lions and tigers, the virus may be using ACE2 receptors or they may use receptors other than ACE2 to gain access to host cells. Lower propensity for binding could translate to lower propensity for infection, or lower ability for the infection to spread in an animal or between animals once established. Because of the potential for animals to contract the novel coronavirus from humans, and vice versa, institutions including the National Zoo and the San Diego Zoo, which both contributed genomic material to the study, have strengthened programs to protect both animals and humans. Zoonotic diseases and how to prevent human-to-animal transmission is not a new challenge to zoos and animal care professionals, said Kotha Klaus-Peter Kerpfli, senior research scientist at Smithsonian Mason School of Conservation and former conservation biologist with the Smithsonian Conserve. This new information allows us to focus our efforts and plan accordingly to keep animals and humans safe. The authors urge caution against overinterpreting the predicted animal risks based on the computational results, noting the actual risks can only be confirmed with additional experimental data. The list of animals can be found here. Research has shown that the immediate ancestor of SARS-CoV-2 likely originated in a species of bat. Bats were found to be at very low risk of contracting the novel coronavirus via their ACE2 receptor, which is consistent with actual experimental data. Please support our channel to grow by pressing subscribe button as well as the bell icon for daily science updates. Thank you.